Theme: Human Embryology, Provisory Organs. Plan. Number 1. Concept on provisory organs. Number 2. Amnion. 3. Yolk sac. Number 4. Allantois. 5. Umbilical cord. Number 6. Chorion. Number 7. Placenta. Provisory organs are organs developing during embryogenesis outside of embryo body. They realize different functions, providing growth and development of embryo. Provisory organs are amnion, yolk sac, allantois, chorion, placenta. Amnion. Amnion is water medium participating in producing of amniotic fluid. It is derived during the second stage of gastrulation as amniotic vesicle composing the epiblast. The wall of amniotic vesicle consists of provisory ectoderm and provisory mesenchyme or mesoderm. Functions of amnion. Amniotic fluid serves as a shock absorber for the fetus, helps regulate fetal body temperature, helps prevent the fetus from drying out, and prevents adhesions between the skin of the fetus and surrounding tissues. Amnion also realizes protective function, preventing passing of harmful agents into fetus. Amnion usually ruptures just before birth. It and its fluid constitute the bag of waters. Embryonic cells are normally sloughed off into amniotic fluid. They can be examined in a procedure called amniocentesis, which involves withdrawing some of the amniotic fluid that bathes the developing fetus and analyzing the fetal cells and dissolved substances. Yolk sac. Yolk sac is provisory organ depositing nutritive substances necessary for development of embryo. It is derived from hypoblast during gastrulation as well of yolk vesicle consisting of provisory endoderm and provisory mesoderm. Functions of yolk sac. It realizes trophic function till 3rd week, supplies nutrients to the embryo. Its basic role is hemopoietic till 7th, 8th weeks of development. It is source of blood cells. In the wall of yolk sac, primary sex cells, gonoblasts, are formed. Allantois. Allantois represents small finger-shaped projection of endoderm in caudal part of embryo. The wall of allantois consists of provisory endoderm and provisory mesoderm. In human organism, allantois doesn't reach great development, but it is organ of gas exchange and excretion. On the 2nd month of embryogenesis, allantois is reduced and forms a part of umbilical cord. It is associated with the development of the urinary bladder. Umbilical cord. It is elastic band connecting embryo with placenta. Umbilical cord is covered by amniotic layer which surrounds mucous connective tissue with blood vessels. Blood vessels are 2 umbilical arteries and 1 vein. And remains of allantois and yolk sac also are present in umbilical cord. Mucous tissue or embryonic tissue gets name of Wharton's jelly. It prevents umbilical vessels from pressing, providing uninterrupted supply of embryo by nutritive substances and oxygen. Chorion. The extraembryonic mesoderm together with the 2 layers of the trophoblast together form the chorion. The chorion surrounds the embryo and later the fetus. In this slide it is imaged by letter C. Functions of chorion. Eventually it becomes the principal embryonic part of the placenta, the structure for exchange of materials between mother and fetus. Functions of chorion. The chorion also protects the embryo and fetus from the immune responses of the mother in two ways. It secretes proteins that block antibody production by the mother, and it promotes the production of T lymphocytes that suppress the normal immune response in the uterus. Finally, the chorion produces human chorionic gonadotropin, an important hormone of pregnancy. Human chorionic gonadotropin stimulates the corpus luteum to continue production of progesterone, necessary to prevent menstruation and for the continued attachment of the embryo and fetus to the lining of the uterus. By the 8th day after fertilization, human chorionic gonadotropin can be detected in the blood and urine of a pregnant woman. During the 4th and 5th months, the human chorionic gonadotropin level decreases sharply and the levels off until childbirth. Placenta. It is provisory organ realizing connection of embryo with maternal organism. It is derived on the 3rd week of embryogenesis from chorion and is finished to form on 3rd month. Villi of chorion in placenta are located in lacuna with maternal blood. Human placenta is hemochorial. Placenta consists of fetal and maternal parts. Fetal part includes chorionic plate, chorionic villi, and amniotic layer. Maternal part is formed by the decidua basalis of the endometrium, which includes basal lamina and lacuna. Between the chorionic and decidual plates are the intervillous spaces, which are filled with maternal blood. They are derived from lacuna in the syncytiotrophoblast and aligned with syncytium of fetal origin. The villous trees grow into the intervillous blood lakes. Cotyledon. Cotyledon, it is the structural functional unit of the placenta. Cotyledon represents stem villus and its secondary and tertiary branches. When the placenta is viewed from the maternal side, slightly bulging areas, the cotyledons covered by a thin layer of decidua basalis, are clearly recognizable. Grooves between the cotyledons are formed by decidual septa. General number of cotyledons in placenta can reach 200. Functions of placenta. Placenta realize respiratory, trophic, excretory, endocrine, protective function. Respiratory function means exchange of O2 and CO2. Trophic function is exchange of nutrients and electrolytes. Excretory function is realized for fetus. Endocrine function is realized by chorial epithelium producing human chorionic gonadotropin, chorionic somatomammatropin, progesterone, and estrogens, predominantly estriol. Protective function of placenta. Immunological competence begins to develop late in the first trimester, by which time the fetus makes all of the components of complement. Immunoglobulins consist almost entirely of maternal immunoglobulin G, which begins to be transported from mother to fetus at approximately 14 weeks. In this manner, the fetus gains passive immunity against various infectious diseases. Placental barrier. It is barrier between maternal blood in lacuna and fetal blood in vessels of villi. Placental barrier includes wall of hemocapillaris of villi, embryonic connective tissue surrounding vessels, cytotrophoblast, and syncytiotrophoblast. Line of toxic substances and most microorganisms cannot pass through the placental barrier. However, certain viruses, such as those that cause AIDS, German measles, chickenpox, rubella, cytomegalovirus, Coxsackie, variola, varicella, encephalitis, and poliomyelitis, can cross the placenta. Many drugs, alcohol, and some substances that can cause birth defects also pass freely. After birth of the baby, placenta detaches from the uterus and is therefore termed the afterbirth. Thank you for attention.